Type O. Type O blood offers the strongest natural protection against several cardiovascular diseases and is the most common blood type worldwide. People with type O have the lowest risk of developing heart attacks, strokes, and blood clots in the legs and lungs compared to all other blood types, a protection that likely stems from naturally lower levels of certain clotting factors, those proteins that cause blood to coagulate. Studies suggest type O individuals may live longer on average because of this significantly reduced cardiovascular risk. Beyond the heart, type O carries significant advantage against malaria, offering roughly 20 to 40% better odds against severe infection compared to type A blood when dealing with the most dangerous parasite strain. Type O provides protection from stomach cancer, which researchers attribute to lower susceptibility to H. pylori infections, a bacterium that can damage stomach tissue. However, type O comes with notable disadvantages. People with type O experience approximately 10 to 15% higher vulnerability to excessive bleeding, particularly during surgery, childbirth, or menstruation. This biological trade-off means the advantage against clotting becomes a liability when rapid blood coagulation is needed. Type O individuals also face higher rates of cholera, plague, tuberculosis, and mumps compared to other blood types, suggesting their genetics may create favorable conditions for certain pathogenic organisms. Despite infectious disease vulnerabilities, type O negative blood holds universal advantage in transfusions, as only 7% of US population carries this rare type, making those individuals valuable donors. Type A. Type A blood creates a unique health profile marked by distinct vulnerabilities in multiple domains. People with type A are significantly more prone to developing stomach cancer, a risk researchers believe relates to higher susceptibility to H. pylori infection, a bacterium that can damage the stomach lining. Type A individuals also face elevated risks of pancreatic cancer and type 2 diabetes compared to type O, though scientists remain uncertain about exact mechanisms. Type A blood increases cardiovascular risk substantially, with people being approximately 50% more likely to develop deep vein thrombosis compared to type O blood. This elevated risk stems from higher circulating levels of clotting factors like factor VIII and von Willebrand factor that promote blood coagulation. From an infection standpoint, type A shows increased susceptibility to smallpox and Pseudomonas aeruginosa infections, as well as higher incidence of cholera and tuberculosis compared to type O blood. One advantage of type A blood lies in transfusion compatibility, as type A individuals can receive from type O and type A donors, and donate to type A and AB recipients. The overall health picture suggests type A requires vigilance regarding cancer screening and cardiovascular monitoring, though lifestyle factors like diet and exercise remain more influential than blood type alone in determining actual disease outcomes. Type B. Type B blood presents a complex profile with distinct disadvantages across multiple health domains. People with type B face substantially elevated diabetes risk, with B-positive individuals at 35% increased risk of developing type 2 diabetes, making it the highest risk blood type for this metabolic condition. Research suggests that type B antigens may interact with glucose metabolism in ways that increase disease vulnerability. Type B also shows increased vulnerability to pancreatic cancer alongside type A, suggesting shared mechanisms related to these blood type antigens. Cardiovascular risks remain elevated for type B individuals, with approximately 50% higher likelihood of developing deep vein thrombosis compared to type O blood, due to elevated clotting factors similar to type A. From an infection perspective, type B shows particular vulnerability to gonorrhea, tuberculosis, and multiple bacterial infections, including Streptococcus pneumonia, E. coli, and Salmonella. These increased infection susceptibilities suggest type B antigens may provide favorable binding sites for certain pathogens. Type B individuals can receive blood from type O and type B donors in transfusion scenarios. The overall health trajectory suggests type B warrants particular attention to diabetes prevention through dietary choices and regular screening, as well as cardiovascular health monitoring. Like all blood types, individual outcomes depend far more on personal lifestyle choices than on blood type classification alone. Type AB. Type AB blood represents the rarest ABO type, present in only 3 to 4% of the population, and carries one of the most concerning health profiles among all blood types. People with type AB demonstrate the highest stroke risk of any blood type due to elevated levels of clotting factors that make their blood more prone to forming dangerous clots within blood vessels. Type AB shows significantly increased risk of cognitive impairment, including memory problems and dementia, with research demonstrating this vulnerability occurs independently of geographic region, age, race, or gender. These cognitive risks trace to higher levels of plasma coagulant glycoproteins like von Willebrand factor and factor VIII, the same proteins responsible for increased stroke and clotting risks. Type AB also faces elevated pancreatic cancer risk similar to type A and type B, and shows particular susceptibility to smallpox, E. coli, and salmonella infections. Type AB individuals lack natural protective antibodies against pathogens carrying A or B antigens, making them more vulnerable to infections that other blood types successfully resist through innate immune defenses. Despite these significant disadvantages, type AB offers one remarkable advantage. AB-positive individuals can receive blood from any of the 8 blood types, making them universal recipients in emergency transfusions. However, the widespread cardiovascular, cognitive, and infection vulnerabilities suggest type AB requires particularly diligent health monitoring across multiple domains. Rh factor. The Rh factor, represented as the plus or minus sign in blood type designations, like O+ or AB-, determines whether a specific protein exists on the surface of red blood cells. If your blood cells carry the Rh protein, you have a positive blood type. If this protein is absent, you have a negative blood type. Approximately 94% of the global population carries the positive factor, while only 6% have negative blood, making Rh negative relatively uncommon worldwide. The primary health significance of the Rh factor emerges during pregnancy, when incompatibility exists between mother and baby. If a woman is Rh negative and carries an Rh positive baby, their blood can mix during pregnancy or delivery, triggering the mother's immune system to produce antibodies against the Rh factor. These antibodies, once produced, can cross the placenta and attack the developing baby's red blood cells, causing a condition called Rh incompatibility or hemolytic disease of the newborn. This potentially results in severe anemia, jaundice, brain damage, or even fetal death in extreme cases. Fortunately, modern medicine has developed immunoglobulin treatments that prevent Rh negative mothers from developing these dangerous antibodies during pregnancy, effectively eliminating this once serious complication in most developed nations. The Rh factor also influences transfusion compatibility, though less critically than ABO antigens. Positive individuals can donate to positive recipients and receive from positive or O positive donors. Negative individuals can receive only from negative donors, but can donate to both positive and negative recipients in emergencies, making negative blood donors particularly valuable. Blood type and personality. Despite widespread popular belief, particularly in Japanese culture where blood type personality theories hold significant cultural presence, there is absolutely no scientific relationship between your blood type and your personality. This association, while deeply embedded in certain cultural traditions, has been thoroughly debunked by rigorous scientific research and fails to withstand empirical scrutiny. A large-scale 2014 survey examining over 10,000 people from both Japan and the United States found that blood type explained less than 0.3% of total personality variance, demonstrating the complete non-relevance of blood type for determining personality traits. Scientists acknowledge that personality likely contains some genetic component, but blood type plays no role whatsoever in that genetic influence. Instead, personality appears molded by experiences, environments, and events encountered throughout life, suggesting external forces matter far more than biological markers. The scientific community generally dismisses blood type personality theories as superstition or pseudoscience, due to complete lack of supporting evidence. While blood type definitely influences physiological factors from clotting risk to infection, susceptibility to disease vulnerability, the idea that it determines whether someone is extroverted, conscientious, or agreeable remains entirely unsupported by evidence. Blood type influences genuine biological mechanisms affecting disease and health outcomes, but does not extend into behavioral or psychological domains. Have you noticed any health advantages or disadvantages linked to your own blood type? Let us know in the comments and subscribe for more fascinating videos about human biology just like this one.